We are Team Eureka.
I'm Vivek.
I'm Zhijie. We are all Secondary 1 pupils from Raffles Institution.
Well, today we are going to demonstrate the DNA extraction experiment, a very simple experiment which requires only common items found in the kitchen.
Okay, but before we start our experiment, we would like to ask one very important question. What is DNA? DNA is actually a nucleic acid that contains the genetic instructions used in the development and functioning of all known living organisms and some viruses.
Simply speaking, the main role of DNA in us is to store information and instructions that make us who we are. DNA is something so complicated, and yet we can simply extract DNA in our own kitchen? Is that even possible?
Actually, DNA extraction is only a simple routine procedure for scientists to collect DNA for research and analysis.
In fact, Friedrich Miescher found DNA in his own lab using this method in 1869. But how did they do that when the meaning of DNA was only found 50 years ago with such advanced technology?
Also, technology in the 19th century had only half of what the 21st century provides. But if we consider the fact that this experiment only needs common household items, it explains why someone was able to extract DNA all the way back in 1869.
So, now that you know what DNA is, it's time we begin the experiment.
You will need a Ziploc bag containing some water, 3 strawberries, a disposable chopstick, about 5 milliliters of liquid detergent, a test tube or small cup, and some cold rubbing alcohol, which is easily found in pharmacies with a 70 to 95% concentration.
First, put the 3 strawberries into the Ziploc bag and squash them until they form a thick liquid. After about 2 minutes, you'll get something like this, a thick liquid. Pour the liquid into the small cup. Next, add some liquid detergent into the cup and use the disposable chopstick to stir for about 20 seconds. Lastly, tilt the small cup and pour the alcohol in slowly and carefully. Alcohol has already been poured into the cup. You will observe that the alcohol floats on the strawberry liquid. After waiting for about 20 seconds for the layers to settle down, you see white stringy stuff, which are actually clumps of tangled DNA molecules appearing in the alcohol layer.
Strawberry's DNA is found in the nuclei of the cell. However, surrounding each cell is the cell membrane. Thus, you will have to break through it in order for the DNA to be extracted. By squashing the strawberries, you have broken open the cells of the strawberries, this process being known as cell disruption.
Adding liquid detergent would melt away the membrane lipids and release the DNA found in the strawberry cells.
Cold alcohol is added to form a white precipitation of DNA. Since DNA is insoluble in alcohol, a solidified strand of DNA forms, which is actually the white stringy stuff that appears in the alcohol layer.
And that will be the end of our science busking demonstration. Thank you.